Welcome, everyone. On behalf of the American Committee and the Weizmann Institute, thank you for tuning in and participating. For those of you who do not know me, I am Dave Donison, the CEO of the American Committee for the Weizmann Institute of Science. We are very excited to host you all today. So, the Weizmann Institute has always allowed the brightest minds to tackle and explore the deepest and most pressing scientific questions, and the current crisis has illuminated just how much of this original DNA has been conserved over the last 70 years. Organically, in just over about 3 months, the Institute has responded to the coronavirus pandemic by launching 60 separate scientific projects. Yes, 60. That's an average of about 5 per week during this period. And to describe in broad strokes some of these efforts, as well as diving deeper into one particularly promising avenue, we are joined by 2 scientists from the Weizmann Institute this afternoon who can detail for us the Institute's international and national leadership in scientific excellence and research. Our main speaker will be Professor Nir London, Department of Organic Chemistry, and we are also joined by the Vice President of the Institute, Professor Roee Ozeri, who is helping to lead and navigate this quickly evolving situation in Rehovot. Professor Roee Ozeri was born in Israel and received a Bachelor's in Science and Physics from the Hebrew University of Jerusalem. He received his master's and PhD degrees in physics from the Weizmann Institute of Science, and he conducted his postdoctoral research at the National Institute of Standards and Technology in Boulder, Colorado, in the group of David Weizmann, the 2012 Physics Nobel laureate. He joined the Weizmann Institute faculty in 2007. So I will ask Roee to make some comments now.
Thank you very much, Dave. And thank you all for joining us tonight. I'd like to use this opportunity to thank you for being longtime supporters and friends of the Weizmann Institute. This means a lot to us always, and in particular at a time like this. So I can tell you a little bit about the status of the Weizmann Institute these days. When the country is slowly, or actually even a little bit quickly, getting out of lockdown. In Israel, schools are slowly going back into action. Commercial stores are going back, and we at the Weizmann Institute are now allowed to work actually at full capacity. We have what we call in Israel a purple badge. A purple badge means that we're a business which is sufficiently needed in order for people to work at full capacity. And that means that at the Weizmann, we're trying to go back into, on top of performing research and directing efforts towards COVID-19, as you'll soon hear, we're trying to direct our efforts a little bit back into normality. And today I got an email from Ayelet, who's here, on next year's budget, which means that things are slowly going back to normal. Still, we have, like anywhere in Israel, we have restrictions about the number of people we can accommodate in each room and the conditions under which more than one person can work in a room. We need separations, perspex glass separations. We need to wear masks, so it's not life as it used to be, but still we're trying to get back on our feet. I know the situation in the US is very diverse and very different in different places, and I hope that quickly in the US things would move back as close as possible to normal. As Dave mentioned, very quickly after probably end of February or early March, different groups at the Weizmann Institute have embarked on efforts to try and direct our expertise and scientist talents towards finding a remedy to COVID-19. And indeed, we have, I think 2 weeks was the last time we counted the number of different projects we have. We have 66 different projects on COVID-19. And these covered a wide range of topics, from different tests, both genetic as well as serological tests, through using AI tools in order to identify outbreaks or come up with computational tools that would find clever exit strategies. Actually, today at the New York Times, there was an opinion piece written by two of our brightest scientists, Ron Milo and Uri Alon, about their rather clever exit strategy, which tries to find a weak spot in the behavior of the virus and synchronize the work cycle of people with the cycle of the virus in order to enable the economy to get back on its feet. Large efforts are directed towards trying to find either vaccines, which would be the best way out, or drugs, as you'll hear from Nir in a few minutes, drugs that would alleviate the danger of the COVID-19 virus. And finally, we also have initiatives that are trying to give tools for kids that are stuck at home to still consume scientific content, get their scientific education, and most importantly, learn about the virus and the COVID-19 disease, believing that information really empowers people and enables kids as well as adults to cope with the situation in a better informed way. So to really give all the resources needed to conduct all these research initiatives, we initiated the Coronavirus Response Fund. And I know that many of you have contributed to this response fund, so I'd like to take this opportunity to thank you for your generosity. The research you'll soon hear from Nir is largely supported by this Corona Response Fund. So I think it's a great way for us and you partnering up and trying to make a difference in this difficult situation. We now understand that it's not a short episode, and we need to look forward, and we need to find the ways to cope either with COVID-19 or with contagious diseases in the longer term. And that's why we're now trying to find ways to establish long-term plans to fight contagious diseases, to establish a contagious disease institute, to look ahead and find ways to cope with the mental difficulties that a situation like this brings through our expertise in neuroscience and psychiatry. So we're trying to look beyond this episode. We're trying to look forward and find longer term solutions to this situation. So with that, I would like to turn and introduce today's speaker. So today's speaker is Professor Nir London. Despite his English family name, Nir is born in Israel, so he's fully Israeli. He did all his academic degrees at the Hebrew University, and then after 4 years of postdoc in the University of California of San Francisco, returned to the Weizmann Institute in 2015, and he's the incumbent of the Alan and Lorraine Fisher Career Development Chair. Nir is an expert in biochemistry. He's an expert on computational tools and the experimental means to test the chemicals that he designs. His expertise, as you'll hear soon, is trying to identify the chemicals that can inhibit different biological molecules, viruses included. And by designing these chemicals, he can search for drugs. So drug discovery is among the main things that Nir is focusing on. And very shortly after Nir realized that the COVID-19 is a pandemic, it's a global situation, he teamed up with an international team of bright scientists to try and find a COVID moonshot, try and find a drug that would remedy COVID-19 in a very short period of time. So please, Nir, the stage is yours.
Thank you, Roy, and thanks everyone for joining. It's an honor and pleasure to describe our research to you. Let me share my screen. Here we go. I hope everyone sees the bright orange introduction slide. So as Roy mentioned, we very soon after the pandemic broke, we established what we now call the COVID Moonshot Consortium, which is a global consortium to try and develop an antiviral drug against COVID-19, but actually more in general against any coronavirus. And we think this is a very important aspect of this effort, as I'll elaborate in a minute. So probably no one needs introduction to the COVID-19 pandemic. But just to give you context, so far, about 4.2 million patients confirmed with the disease, which led to almost 300,000 deaths. This gives you really the magnitude of how important it is to solve not only COVID-19, but the next SARS-related epidemic that is probably imminent. This is already the third outbreak from coronaviruses that the world has witnessed. We might had others that were silent epidemics, and we maybe already have immunity to them, but there's a wide consensus that we may have additional coronavirus epidemics. So it's really important to find a drug, not only to tackle this problem, but the next one as well. So globally, there are two main strategies to fight viruses. One is vaccines, which were hugely beneficial and drove out diseases like human papillomavirus, mumps, measles, and rubella. And these are indeed very good strategy to fight viruses. However, I want to point your attention to the fact that vaccines are expensive. They have a lot of logistical problem. Typically, they need refrigeration. Their manufacturing is difficult. It's hard to distribute them. And typically, vaccine solves first world problems. Whereas the second strategy to fight viruses are small molecule antiviral drugs. Some successful examples include HIV drugs, hepatitis, both B and C, herpes, and cytomegalovirus, which for all of them and successful antivirals were developed. These are typically cheaper to produce, to distribute. You can spread them globally very fast and efficiently. And so we're in this project, we intend to develop an antiviral for coronaviruses. So our goal is to develop a cheap and safe antiviral against a specific protein, which I'll introduce in a moment. And we'll try to do it much faster than the traditional drug discovery pipeline. And I'll outline our approach to try and achieve that. But most importantly, we think that antiviral and anti-infective drug discovery is problematic. The current way pharmaceutical address it, it's problematic because it's not commercially viable. So developing the next antibiotic will not make a lot of money for big pharmaceutical companies. Another good example is as soon as the first SARS epidemic died down, all drug discovery efforts against SARS immediately stopped because there were no patients, there were no one to buy the drugs, and then no one progressed and actually brought something to the clinic. Had we had the foresight in 2003 to keep on pushing and take the drugs against SARS to the clinic, we would have no problem with COVID-19. So we think that a new approach is needed where the investment in health should be decoupled from commercial interest, and we think global collaboration and crowdsourcing might be one solution to this problematic incentives in the discovery of antivirals. Just to very briefly introduce the causative agent for COVID-19, which is this SARS coronavirus 2. You can see here an electron microscope figure of the virus and the famous crown or corona for which it is named. A few numbers about the virus from Ron Milot's recently published paper. It contains only 30,000 RNA bases, which is less than 0.001% of the human genome. And these encode less than 30 proteins. So all of this havoc is wreaked by only 30 tiny proteins. It takes on average 10 minutes to enter a cell, and once it enters, it starts to replicate itself. And after about 10 hours, 1,000 new virions erupt from the cells and go on to infect neighboring cells. And here you can see a schematic of this life cycle. So it enters the cell, it replicates itself, and then it buds out and infects new cells. And our target is this process over here. So after entering the cell, the virus expresses two very long proteins which needs to be processed, to be cut. They need scissors to cut them into functioning units. So here it's described by these long lines are going into these circles. And this here is the protein that's responsible for processing the long protein into the functional protein. It's called a protease. And it's a molecular scissor that processes the long proteins into the functional proteins. And if we find a small molecule here depicted in yellow circles that would latch onto this protein and inhibit it from performing its function, then we'll find a drug that inhibits viral replication, and virions, even after the person is infected, virions would no longer be able to replicate and to destroy additional human cells. So that's our target. Who are we? So our team at the Weizmann Institute has, as Roy described, technologies to discover a specific subgroup of drugs called covalent drugs. These are drugs that are irreversible. They're able to bind to their target protein irreversibly, and so they have much longer duration of action. They're more potent. Typically, in the drug discovery industry, these types of drugs are aimed for oncology indications, and indeed that was our main research focus before Corona erupted. But this specific protease, this protein that I just showed you, is very, very suitable for the exact same technology. So its active site contains a chemical moiety that we can target with our technologies. And that's why we really very rapidly started our efforts against this protein. Other key players are a group at Oxford and Diamond Light Source in the UK, which are able to determine crystal structures of the protein, so they can take photos at atomic resolution showing exactly where every atom in the protein is found and exactly how our drugs bind to the protein. I'll show you some of these photos in a couple of slides. Another key player is Enamine, which is a very large chemical vendor or contract research organization. Enamine agreed to synthesize any compound that we propose to them basically at cost. So currently they're devoting 15 chemists to this project, and these are working full-time just synthesizing compounds that we are designing and then shipping them for testing. Other partners, such as the University of British Columbia, are testing compounds. A lot of the testing, as I'll describe, is also done at the Weizmann nowadays. Teams at Memorial Sloan Kettering are doing drug binding simulations, trying to prioritize which compounds might work best. A biotech in California called Postera helps us with applying machine learning algorithms to design routes, telling us how to chemically synthesize these compounds. And other groups are helping with viral assays and with medicinal chemistry support. And I'll describe some of this process in a minute. So our approach is to rely on several force multiplier. The first is crowdsourcing the global intelligence. One key aspect of this project is that it's completely open science. So there's no patents, there's no intellectual property. All of the progress in the project is published online immediately, as soon as technically feasible, to share with the scientific and the public, scientific community and the public. And indeed, so far, we generated an unprecedented amount of preliminary data which I'll show you in a minute. And then the last sort of peg we rely on is machine learning and crowd computing. So these drug binding simulation, for instance, that I mentioned, are ran on Folding at Home, which is a crowd computing platform in which personal computers are being donated. Their compute time, when you go to sleep at night, but you leave your computer open, it can run calculations for us to try and assess different drug binding to this target. And so far, so many people have contributed their personal computers that it actually, in aggregate, is now the strongest computer in the world by far. So it more than 10 times than all other supercomputers combined. So this has been tremendous. And actually all of the really different people and different labs recruiting to fight this pandemic was really nice and inspiring to see from a scientific perspective. So this project started around mid-February, when the team in Oxford were able to clone and produce the protein, the main protease. They're not working with the live virus, but they're working with our target protein. So they were able to produce it in bacteria. And then within a week, they were able to solve its atomic resolution structure. So here you see a representation of how the protein actually looked like, even though it's a tiny, tiny thing, about 30 nanometers in width. In parallel, they shipped us a sample of that protein to the Weizmann Institute, where we were able to apply our technology for discovery of covalent inhibitor for these irreversible inhibitors that I described to you. And we ran our screening platform and were able to identify about 150 active sites hits. These are a starting point from which we can start to develop a drug. We let them know they have a copy of this entire library, so they could very rapidly try and now determine new structures of the protein, but now in complex with our drug. So they could tell us, here is how they bind based on these binding structures. So here you can see many, many different fragments bound in the active site of the protein. Now we can take this data and try to design the next generation of more potent and more effective inhibitors for the protein. So here you see a different view. This is the protein from far away, and this is the active site. And you can see how different fragments, different molecules occupy different sites within the protein. And if we zoom in, you see how they really cover the entire active site, this pocket or crevice over here, almost exhaustively. And now the idea is to take different hits or different structures and try to merge them into something more potent. So you can see several ideas here. So for instance, if the light green fragments turns right and the dark green turns left, we can try and merge them and make the compound that has both heads. And this would likely work much better than each one of them separately. The problem is there are thousands and thousands and thousands of such potential combinations. And this is more than we can single-handedly work out. So Postera, the biotech from San Francisco, opened up a platform for crowdsourcing the medicinal chemists of the world, or any chemists in the world. Basically, they set up this web platform where chemists can design compounds and submit them. And then we offered to synthesize them and test them and see if they actually work. So we gave them the data. We gave them all of the tools, basically, and we asked them to input basically their knowledge and expertise. And we published this on Twitter. This got more than 500 retweets. It spread like wildfire. And very soon we started getting responses from people. This was back when everyone were in shutdown, and people were looking for ways to contribute. So these computational chemists and medicinal chemists with sometimes years of experience from pharmaceutical companies, just starting submitting ideas and insights based on the data we produced. This one is one I especially liked. I just spent the last 72 hours crunching with some other guy to design potential antiviral drugs for the COVID moonshot. We submitted 66 design in total across 3 crystal structures out of the more than 90 crystal structures that we published. So the response was really overwhelming. And here you can see a random selection of designs that were submitted by people, and you can maybe appreciate how diverse they are and how unique and creative people can be in designing these molecules. And so far we got more than 4,500 designs from 250 different designers. And we were actually able to make, so Enamine, our chemical contract research organization partner, as soon as the design starting to pour in, we rank them, we prioritize them, and we send them to synthesis. They were able to make 400 compounds. And only last week, we were able to ramp up our testing capabilities. Here at the Weizmann, we use the high throughput screening facility at the National INCPM. This facility is geared towards testing many, many compounds. And so within a week, they were able to test the first 280 compounds. And we already found potent hits in this first sort of wave of compounds. And in parallel, these compounds are also sent to Oxford, where they try to solve the structures of these new improved compounds, validating the hypothesis based on which the compounds were designed, which places us in a much better place to design the next sort of generation of compounds. And only today we published this activity data. And so here you can see an example of anyone can go to Postera's website, can see the actual designs and the activity data, basically how well does each compound inhibit the target protein. They can search their own design if they submitted one. They can see all the different parameters, you know, how potent the compound is, different physical properties, similar other submissions. They can click on a compound and try to see if the submitter gave the rationale. You know, this is how I designed the compound. I use this and this structures for inspiration, and I use maybe that reaction to build the compound. And so people can comment on other people's suggestions. There's a lively forum where you can start a discussion saying, you know, I liked your design, maybe you'll add this atom, etc. And a community, sort of a scientific community, is built around this platform. And so even though we're only, you know, a handful of groups, where actually we have a personnel of maybe a few hundred people constantly thinking of new designs and new ways to improve our compounds. And we think this is a really big force multiplier. So just two sort of series that I'm more proud of, because these are things that we developed in our lab in parallel to these crowdsourcing ideas. So we also have some experience in designing compound. These are 3 students that in the midst of the pandemic worked day and night to synthesize chemicals that all share this basic scaffold. They have different sort of decoration on this side. And you can see here how compound after compound, they were able to improve the potency. Red is better, and you really can see the gradual improvement, and these compounds are really looking very nice. And all of these results are, of course, also published back to the community and can be combined with other chemical series that are being tested. Here you can see how we show, so if you remember, the compound is irreversible. So this is a mass spectrum of the protein. So basically we weigh it, and you can see this is the mass of the protein without the compound. And when we incubate it with this compound, it shifts, the protein gains weight, basically, and the weight is exactly corresponds to the weight of the compound. So this is how we know we actually labeled the protein irreversibly. A second approach that we pioneered in the lab, Daniel is a computational student who was able to take this green, if you see here, this green drug compound. This was developed against the original SARS, so 17 years ago. But because the enzymes are so similar between the original SARS and this SARS, compounds that hit the original SARS are also active against the new one. So Daniel modified the green compound, added this magenta part here, and predicted this would now be an irreversible inhibitor. Paul, a chemist in the lab, synthesized it, and it actually worked. And now Inamine in the Ukraine are synthesizing this whole matrix of derivatives, and already one of them worked really, really nice against the virus. So this is an example how different approaches can also be incorporated into this consortium. So just to sort of finish up, our goal currently is to get a compound into animal testing within 6 months. So this might not have effect on the current pandemic, since drug discovery is a really long process. Traditional drug discovery takes 10 to 15 years and $1 to $2 billion. We hope to get to a drug much, much faster. But as I mentioned before, our real aim is to prevent future pandemics, because this enzyme is very, very highly conserved across all coronaviruses. It's more than 95% identical, in fact, to the previous two outbreaks of coronaviruses. We think that if we have a drug against this protein, it will also be effective for future pandemics, and it will be our insurance to not be in the situation again. So currently, we're trying to ramp up our chemistry capacity. As I told you, Inamine were able to provide 15 chemists. A CRO in India contributed 5 chemists. And we're contracting another company in China, hoping to get another 20 chemists. The more chemists we get, the more compounds we can make, and the faster we can reach our clinical candidate. And we're predicting that next month we already have potent enough compounds to test them against live viruses through various collaborators. And if we talk again, I hope I'll be able to update you with candidates that were able to kill the live virus. That's it. I want to thank you for your attention. I'll be happy to take any questions you may have.
So the first question I have for you, Nir, is, if you have any obstacles to your path right now being successful, what are they? And I guess another follow-up question would be, what could accelerate your efforts? What could make this happen even faster?
Right. So as I alluded to in the final slide, currently we have a lot of ideas of how to go. We have the infrastructure to test them. And the bottleneck is chemistry, synthesizing the compound. Synthesizing a single compound can take 2 weeks. If it's complicated, it can take a month. If we want to move forward as fast as possible, we need to make as many compounds as possible in parallel. And this requires more and more chemists. So for now, we're maximizing our sort of funding capacity. We hired all the chemists we could fund. If we had more funding, we can fund more chemists. So there are companies in India and in China that very, very good companies, credible and reliable, that have thousands of people ready to hire. So some of them were actually contributed, freely, pro bono, but some of them still insist on getting paid. And even those that work pro bono need to recoup their costs. So the funding basically directly translates to more chemists making more compounds in parallel.
So Nir, as a follow-up question, somebody asked, as a scientist, what has been the most surprising thing that you've learned, or things that you've learned along the way? What has been the most, I guess, unexpected finding?
So I have to say I was a little skeptical about the crowdsourcing idea. I was sure that you let everyone voice their opinion, you'll get crappy opinions. And I was pleasantly surprised that, so currently we're sort of, we have 3 or 4 main directions based on the data we already collected. And one of the most promising, one of these 3 or 4 directions actually came from a crowdsourced idea. So I was very pleasantly surprised to see that it's not all garbage, what the global community has to and this actually made us really want to wade deeper into the list of suggestions we got from the crowd, because if there was one gem in it, maybe there are others.
So I think that when we have someone as knowledgeable as you, these are the kind of questions I was expecting, which would be, what about going after repurposing already approved drugs?
Right. I think that's a great idea to explore. I think many groups are trying to do that. I'm a little pessimistic since I'm not aware in maybe 20 years that this concept of repurposing has been around of a successful story. There's one successful story of it's actually kind of tragic, thalidomide, which was originally designed for morning sickness, but was teratogenic and caused really horrible results in newborns. Was years later was repurposed as an anti-cancer drug, and it's now hugely successful as anti-cancer drug. But other than that, I'm not aware of any successful repurposing effort. Despite that, people are trying. People are screening, these libraries of approved drug or safe drugs. And I'm sure these are small libraries. There are not so many approved drugs. So I'm sure if something is out there, they'll find it fast.
So I guess as a follow-up, is the first, conditionally approved product is Remdesivir. Are you familiar with that?
Yes, I'm very well familiar.
Can you comment a little bit about that mechanism of action, what its history is?
Right. So Remdesivir was developed as an antiviral for the Ebola virus, which is also an RNA-dependent virus, like corona. So an RNA virus, basically, its genome is RNA, and if I remember correctly, it's Remdesivir is an analog of nucleotide. So it looks like an RNA base. So it basically incorporates itself into the viral replication machinery when it tries to replicate its RNA, and it stops it short. The clinical trials against Remdesivir, although they reached their endpoints, I think were slightly disappointing. So the the results, the improvement seen in treated patients versus untreated patients was, although statistically significant, somewhat underwhelming. I do think that maybe we'll see better results if we try to treat earlier. So the earlier you treat with Remdesivir, the viral load in the patient is lower, and the drug will be have more potency, or it will be more efficacious. So maybe they set up a really high bar. They did the clinical trial against very advanced patients, and they were only able to shave 3 days from their. But maybe as soon as you start showing symptoms, if you have enough Remdesivir to take and it's safe enough, then maybe you won't even get to need a respirator. However, one point that I want to make: Remdesivir is a very complicated drug to produce. As you know, all of the stock basically that Gilead have of Remdesivir, they gave over or donated to the federal government, and they're trying to ramp up production now. So one of our key criteria in developing our drug candidate is to make it a very easy to make compound. So this wouldn't be a bottleneck for creating the number of dosages you need to treat the global population, and wouldn't be, wouldn't translate into only Western countries with production capabilities being able to get access to the drug.
This is a question about a comment that you made earlier in your presentation about the pharmaceutical development market, which is, you talked about uncoupling the market from drug discovery. So I guess what the question is, what would be a better viable system to make sure that we are developing drugs, so if a pandemic like this happens, we have tools, we have products and therapies to use?
So I think for essential drugs that do not have commercial potential, there need to be organizations, whether it's the World Health Organization or governments or philanthropic bodies such as the Bill and Melinda Gates Foundation, or some sort of mechanism to also fund the very costly clinical development of drugs that do not have commercial potential, but are acute for pandemic preparedness for third world countries, etc. And I think there's sort of a void in this niche currently, and maybe this pandemic highlights that need, and maybe, you know, these sorts of initiatives offer an alternative model that would be easier for such body to fund and to support, since no one is going to make any profits off of this effort.
Yeah, so I think this is an interesting follow-up to your answer. You know, obviously this is a very unique and novel approach to designing a therapeutic for all of the reasons that you explained. So what is it about the Weizmann Institute, about the environment and the ecosystem that you're existing in, that lets you carry out your research in such a novel way?
So I think it's the fact, well, two facts. Am I allowed two facts?
You can have three.
First, it's, you know, the Weizmann always encourages curiosity-based science. And we developed our technologies not in order to cure coronaviruses, right? We didn't know this is the acute need that will rise. And the fact that the Weizmann basically let me do whatever interests me to begin with translated into us being ready with this unique technology that was very suitable to attack this particular protein in an emerging pandemic. So that's one basis. The second basis is the infrastructure that the Weizmann provide, which is, I think, unparalleled anywhere else. And to exemplify that, so in the beginning of the project, we had a couple of academic labs that were slated to perform the activity assays of the compounds. So we'll design them, they'll synthesize them, and this and that lab will test the activity of the compounds against the protein. And once we had the first wave of compounds, they realized, well, maybe it's not working as well as we thought. And I decided that this is, at that point, that was a bottleneck, and I want to implement the testing at the Weizmann. And literally within a week, we were able to mobilize this technology platform at the high throughput screening unit and to develop the assay and to run it against hundreds of compounds. And this was only possible because of the existing infrastructure, facilities and services, service units at the Weizmann. So I don't want to make comparisons, but Oxford weren't able to get to that point as fast. So whoever taking notes.
So this is related to another virus that we live with, but I'm sure, as you're well aware and that we've publicized a lot, that Professor Ruth Arnone has been working on a universal flu vaccine that I believe is in phase 3 clinical trials now. Do you think that there'll ever be a universal vaccine or preventative for something like this, for a coronavirus?
I think it's highly possible. I think they're understudied, so we don't know enough to say about them. You know, are they amenable to vaccination or not? And we don't have a vaccine yet, but it's highly likely. They're certainly very, very conserved between different species of coronaviruses are conserved. There are even some talk very recently that people that were exposed to the first SARS are immune to the second SARS. So this bodes well for universal vaccines for coronaviruses. But take this with a grain of salt. I'm not an immunologist. I'm a chemist. So only my opinion from what I read in the scientific literature.
That's great. And that was actually the last question that I had for us today. I did want to just end by letting everyone know that if they have the access to the New York Times, either the physical copy or the digital copy on the opinion page today. Two Weizmann scientists have a fascinating essay that they sent in and got published, and Roe had mentioned that earlier. I want to obviously thank all of you for your questions, your participation. On behalf of all of us, I want to thank you, Nir, and you, Roe, for everything that you do with the other scientists and the students at the Weizmann Institute, and for all your heroic efforts in combating this global pandemic. I wanted to also let everyone know that we're proud at the American Committee to work with the Weizmann Institute advancing a global fundraising drive to raise $25 million to fight the novel coronavirus and to fund those 60 projects that I spoke about. And I'm happy to already announce that because of the generous support of our supporters across the world, and especially the United States, we've already received many millions of dollars for that effort. And for those of you who are interested in getting involved, I encourage you to reach out to your local Aquis professional, or you can email me personally and we can follow up and give you information of how you can also get involved in this effort. So I hope you are all safe, hope you all stay healthy, and I look forward to the next time that we can all get together.
Thanks
Thanks
everyone.
and bye.